My name is Anushka, and my topic is Understanding the Origin of SARS CoV-2 and Comparison of COVID strains and their analysis. The 2019 coronavirus pandemic has impacted the whole world, causing billions of people to become infected and millions of deaths. Several scientists and researchers around the world conducted research that led to the answers of several questions that were unsolved at the outset of the pandemic. A lot of research has been done and is still being done. The virus's ever changing and evolving nature has resulted in a wide range of variations. The World Health Organization has identified certain of these variants as variants of concerns due to their nature and behavior that can be harmful to public health. These worries could stem from their high transmissible character, for example. Different versions have different mutations, which must be recognized accurately in order to build a treatment plan or preventive strategy to combat the virus's propagation. During any therapeutic or preventive research, understanding the changes in the sequence, knowing the mutations, and so on becomes extremely vital and crucial. The origin of coronavirus was a critical topic that needed to be answered before any important study could be conducted. The animal and the geographical location that are related to the virus, the origin of the virus, are referred to as the virus's origin. After figuring out the SARS-CoV-2 origin, the next question is how the virus got to humans and started infecting them. Informatics tools could be utilized extensively to comprehend all of this. Another set of questions that must be answered before any therapeutic or preventive study is what mutations have occurred in the virus and how they have affected the transmission, infectivity, and other aspects of the virus. One of the most recent and dominating mutation is the D614G mutation, which has proved to be responsible for increased infectivity of the virus. In the discipline of bioinformatics, there is a range of tools that can be utilized to do research. This includes softwares such as Eugene for visualizing sequences and observing changes in sequences, Victory for visualizing phylogenetic trees, and databases such as NCBI virus. The goal of the project was about understanding the origin of the virus by comparing different sequences from three organisms, humans, pangolins, and bats, and then retrieving 16 sequences, which included 10 unclassified sequences, one sequence each from the alpha variant, beta variant, gamma variant, delta variant, and omicron variant, and one reference genome, and then analyzing it further to know and understand their lineages and mutational patterns, especially in spike protein. Because the virus's S protein, also known as spike protein, is such an important component of the virus, keeping track of the S protein's changes is crucial. It is important to keep track of the virus's mutational alterations and the variations of concern. Methodology. The first step was phylogenetic tree analysis using T-bioinfo server to know the origin and the evolutionary pattern of the virus. A phylogenetic tree is a very helpful tool that can help understand evolutionary patterns. NCBI virus database was searched for SARS-CoV-2. After that, applying the required refined result feature filters, the geographical location was set to China since the virus originated from there. The nucleotide completedness was set to complete to eliminate any of the partial sequences that would be there in the results, and the host was set to pangolin, bat, and humans. Using the download button on the top of the page, the required database was obtained. In addition to that, the reference sequence was also obtained, which is also available on NCBI virus. Next step was the phylogenetic analysis of the dataset using the T-bioinfo server. On the server, the phylogenetic analysis option was selected. Next, using the options given, the files were uploaded. and 4 groups were made, namely human SARS-CoV-2, bat coronavirus, bat SARS-CoV, and pangolin coronavirus. After uploading all the files, the pipeline was made. The steps that were followed to make the pipeline are showed in the picture. After making the pipeline, it was then ran and named. In the output files section, which was obtained as the result of the pipeline, the median tree was downloaded and was visualized using the fig tree software. The next step was performing multiple sequence alignment, and for the same, the MSA output was obtained. Multiple sequence alignment is also known as MSA and is the alignment of 3 or more biological sequences of equal or similar length. The homology and the evolutionary relations between the sequences that are being examined can be obtained from the output we received. In the case of understanding the origin of coronavirus, multiple sequence alignment was used to obtain the reason why pangolin coronavirus is closer to that of SARS-CoV-2 than to bat coronavirus. To do the same, MSA was performed and then visualized. The visualization of the MSA output obtained from the phylogenetic analysis pipeline was performed using the UniPro Eugene software. For this, multiple sequence alignment output was downloaded from the output files of phylogenetic analysis. Then, using Eugene software, the results were visualized for MSA at the nucleotide level. After visualization of the results, a conclusion was drawn. After multiple sequence alignment at the nucleotide level, a similarity index was obtained using the Eugene software, and based on that index, results were observed and conclusions were drawn. After performing the steps of MSA at the nucleotide level, MSA was performed at the amino acid level to know and understand the sequence differences of SARS-CoV-2 at the amino acid level. For this, the file that contained the data for multiple sequence alignment and at the amino acid level was downloaded from the output files of the analysis. After understanding the origin of SARS-CoV-2 and concluding the results, the next step was to select a dataset and work on that dataset to know the similarities and pangolin lineages of the randomly selected 10 unclassified sequences. The dataset sequence was retrieved from NCBI virus, which included 16 sequences containing one sequence each from alpha variant, beta variant, gamma variant, delta variant, omicron variant, one reference sequence, and 10 unclassified lineage sequences. Next is the phylogenetic analysis of the dataset. After obtaining the sequences from NCBI virus, phylogenetic analysis was performed. On the tBioInfo server, the phylogenetic analysis was selected and the files were uploaded. After uploading the files, the uploaded files were placed in different groups according to the sequences. All the unclassified sequences were placed in one group and the variants were placed in different groups. After uploading all the required files, the pipeline was made. After the processing of the pipeline is completed, from the output files, the phylogenetic tree file was downloaded and visualized using fig tree. The MSA output at nucleotide level was downloaded and was visualized using the UniPro Eugene software, and from the output temp files, the file for MSA at amino acid level was downloaded and visualized using the UniPro Eugene software. The next step was identifying the lineages of the sequences. For the lineage of the unclassified sequences, GISAID was used, whereas for the lineage of the classified sequences, the COV lineages database was used. The GISAID clades can be used to know the pangolin lineage of the unclassified sequences. After that, this next step was identifying the most prevalent mutation among the unclassified sequences. After understanding the pangolin lineages of the sequences, specific mutations were looked into to understand the sequence much more efficiently. This was done using the Pangolin lineage list and outbreak.info. Majorly, the S protein was looked at since it is a key component of the virus that affects its infectivity. Results and discussion. The phylogenetic tree that was visualized using the Fig tree software shows that it is evident that the bat coronavirus emerged first, or in other words, was the earliest one when considering the origin of the coronavirus, and may have mutated to give rise to human SARS-CoV-2 and pangolin coronavirus. The evolutionary relationship between SARS-CoV-2 and pangolins is closer since they are in clades closer to each other. After analyzing the phylogenetic tree obtained using the phylogenetic analysis pipeline, from the same output files, MSA file were downloaded. It was present under the temp files under the phylogenetic output files. The Unipro Eugene software was used to visualize these sequences. The results of the multiple sequence alignment show that in comparison to pangolin coronaviruses and SARS-CoV-2, bat coronaviruses shows higher amount of variations and mutations. The next step that was to quantify the similarities among the sequences. This was done using the similarity index matrix offered by the Eugene software. To understand the similarities between the sequences and to effectively compare them, a quantified data table was obtained using the Eugene software, which had a similarity index of sequences. It was done with respect to reference genome. This similarity index shows that there is an 85% similarity between SARS-CoV-2 and pangolin coronaviruses, whereas the similarity percentage between the sequence of SARS-CoV-2 and bat coronaviruses is in a range of 78% to 79%. This shows that the similarity between the sequences of SARS and pangolin coronavirus is higher than the similarity between the sequences of SARS and bat coronavirus. In other words, pangolin coronavirus is more similar to SARS-CoV-2 than bat coronavirus as per the similarity index as shown by Eugene. This was MSA at the nucleotide level. In the next step, multiple sequence alignment was done of the four groups, namely human SARS-CoV-2, bat coronavirus, bat SARS-CoV-2 and pangolin coronavirus at the amino acid level following similar steps as those of the MSA at the nucleotide level. The sequences were also visualized using the Unipro Eugene software. Unlike the multiple sequence alignment at the nucleotide level, the dissimilarity index was obtained using the Eugene software in the MSA at amino acid level. The results that were obtained from the dissimilarity index shows about 7% to 9% dissimilarity among the pangolin coronavirus and human SARS-CoV-2, whereas the results of the comparison of sequences of bat coronavirus and human SARS-CoV-2 shows a dissimilarity of 15 to 17%. And hence, similar to the conclusions made by MSA at the nucleotide level, SARS-CoV-2 has a greater similarity with the pangolin coronavirus compared to that of the bat coronavirus. Another indication that can be obtained from the results is that not every difference which was at the nucleotide level got translated into the amino acid level. This can be implied because the nucleotide level has a greater percentage of variations than the amino acid level. This refers to that pangolin coronavirus is more comparable and similar to human SARS-CoV-2 than bat coronavirus. Next are the results of the phylogenetic analysis of the dataset sequences. The dataset was obtained and compiled using the NCBI virus database. The phylogenetic tree was obtained using the tBioInfo server phylogenetic analysis pipeline and was visualized using the Fig tree software. The results that were concluded using the phylogenetic tree are that unclassified sequence 9 and unclassified sequence 7 are close to Omicron sequence. Unclassified sequence 1, unclassified sequence 2, unclassified sequence 8, and unclassified sequence 10 are closely related to each other. The group consisting of unclassified sequence 1, 2, 8, and 10 is close to the group consisting of unclassified sequence 9, 7, and Omicron sequence. Unclassified sequence 6 is closely related to unclassified sequence 5 and the gamma variant sequence. Unclassified sequence 4 and the delta sequence are closely related to the alpha sequence. Even though unclassified sequence 3 and the beta sequence are on different branches of the phylogenetic tree but are closely placed to each other. After performing the phylogenetic analysis and the MSA analysis, the next step was to know the Pangolin lineage of the sequences. Firstly, the Pangolin lineage of the variants of concern was obtained. This was done using the Pangolin lineage software. The results that were obtained are that the sequence of variant of concern alpha has lineage as B.1.1.7. The sequence of variant of concern beta has lineage as B.1.351. The sequence of variant of concern delta has lineage as B.1.617.2. The sequence of variant of concern gamma has lineage as P.1. The sequence of variant of concern Omicron has lineage as B.1.1. The sequence of reference genome has lineage as B. The next part was to know the Pangolin lineages of the unclassified sequences. This was done using the GISAID software. According to the results, unclassified sequence 1, 2, 8, and 10 have clade as GRA. Unclassified sequence 3 has clade as GH. Unclassified sequence 4 and 5 have clade as G. Unclassified sequence 6 has clade as GR, whereas unclassified sequences 7 and 9 have clade as other. These clades can help determine the Pangolin lineages of the unclassified sequence since they were not determined using the Pangolin lineage database. Clade GRA has dominant Pangolin lineage as B.1.529 and its descendants BA.1 and BA.2. Clade GR has dominant Pangolin lineage as B.1.1. Clade GH has dominant lineages as B.1.113 and B.1.351. Clade G has dominant lineage as B.1.80 and B.1. The clade which shows as other refers to that they don't match the required criteria which the other clades do. Final conclusions that can be drawn are unclassified sequence 1, 2, 8, and 10 have the lineage as that of Omicron and its descendants BA.1 and BA.2. Unclassified sequence 3 has the lineage as B.1.113. Unclassified sequence 4 and unclassified sequence 5 have the dominant lineages as B.1. Unclassified sequence 6 has the Pangolin lineage as B.1.1, whereas unclassified sequence 7 and unclassified sequence 9 don't match the criteria the other sequences do and hence have been marked as other, but according to the phylogenetic tree analysis, they are closer to the Omicron variant. Next are the results of mutation analysis using outbreak.info. For different lineages that are related to specific clades, mutations can be tracked and observed using outbreak.info database. The most prevalent mutation that can be observed in the lineages is D614G in locations where SARS CoV2 isolates Expressing a D614G mutation as seen above, the viral spike S protein prevails for some time, indicating that the D614G modification increases the transmission of the virus and its infectivity. Coronavirus is a group of different viruses including SARS-CoV-2, which is responsible for the COVID-19 pandemic. Coronavirus is a single-strand RNA virus, which refers to the fact that the genetic information of coronavirus is encoded in its RNA. Understanding the origin and identity of a virus is a very crucial step to initiate appropriate research for both curative and preventive research. This would help understand the origin of COVID-19, to understand the jump of the virus and the sequential changes that led to the virus to start infecting human beings, to understand the evolutionary relationships, and many other questions. Phylogenetic analysis and other methods were incorporated into the project. From the phylogenetic and MSA analysis, it is evident that the bat coronavirus evolved initially or was the original coronavirus, and when examining the origin of coronaviruses, and that it may have mutated to give rise to the human SARS-CoV-2 and pangolin coronavirus. Human SARS-CoV-2 and pangolin coronavirus have a closer evolutionary relation since they are in clades that are more closely related or placed to each other. The bat coronavirus, in comparison to that of SARS-CoV-2 and pangolin coronavirus, has more mutations. This shows the organism that might have been responsible for the virus spreading to human beings and causing a pandemic. These results were used to conclude that human SARS-CoV-2 and other has more similarities with pangolin coronavirus than to bat coronavirus. Mutations in a viral strain can bring a variety of changes ranging from reduced infectivity to increasing its infectivity many folds. And hence it comes very importantly to understand the origin and mutations to understand a virus. In this project, the pangolin lineage of the different sequences was analyzed to further understand the most common mutation out of the 10 unclassified sequences. Pangolin, which stands for Phylogenetic Assignment of Named Global Outbreak, is a tool for the implementation of the nomenclature of the lineages of SARS-CoV-2. The lineage was identified using two databases, namely GISAID and Pangolin lineage. The two databases helped identify the lineages of the unclassified sequences and the classified sequences. After the phylogenetic and MSA analysis, results were drawn and it was identified which sequence is closer in evolutionary patterns to which sequence. After getting to know the most common mutation, it was easier to understand the sequences and the virus. The most common was the D614G mutation. The D614G mutation has proved to increase and enhance the infectivity of the virus. This point mutation has taken over others in terms of prevalence. Among the sequences that were selected for the project showed that most common was the D614G mutation. Thank you.